Okay, now that we know the structure of the DNA molecule and we understand that the DNA molecules are replicated, so the daughter cells have the necessary information that they need to function at the end of mitosis or meiosis, it's now really time for us to address how the cell uses that information to build cellular structures, primarily macromolecules, which then can be used to build a variety of different parts of the cell. And so this leads us to the discussion of gene expression. How do you go from a gene to an actual protein that has a function on the inside of the cell? All right, so just a little bit of review. We know that the way that genetic information is stored in genes is in the specific sequence of nucleotides, A's, T's, C's, and G's. Every single gene has a different order and a different length to how many nucleotides are put together in that sort of chunk of information. And we also know that DNA is inherited by an organism, right, from the previous cell or from the previous generation, and the DNA is going to dictate these specific traits. Now, the DNA itself doesn't, let's say, directly lead to or cause these specific traits, but it holds the information that creates the proteins, and it's really the proteins that are going to lead to these specific traits that you see in an organism. So, for example, you have your mom's freckles. We have probably used this analogy before, right? Your mom didn't just give you a little satchel of freckles when you were born and say, here you go, here are some of my freckles for you, right? Your mom instead gave you or passed on to you her DNA, her genes, her genetic information. And somewhere in her DNA, there was this gene that coded for an enzyme, a protein that has the ability to create pigment and put that pigment into the surface of your skin, giving you the appearance of freckles. So really, this enzyme, this protein, is the link between your genotype and your phenotype, right? It is the link between the genetic information that's coded in your DNA and the actual outward expression of that information. And so we have to talk about how do we go from DNA then to creating this protein. And this process is called gene expression, and it happens in two stages. The first stage is called transcription, and the second stage is called translation. And what we're going to do first is we're going to go through a little bit of background in both of these stages before we go into more detail on one stage individually. And so transcription, first of all, is the creation of an RNA molecule using the information that's coded in the DNA. RNA, so let's say DNA is not turned into RNA. DNA is simply read, and the information that's read in the DNA is used to make a new RNA molecule. Transcription produces a very specific kind of RNA molecule. This is called a messenger RNA or an mRNA. You can think of it as this is the molecule that carries the message from the DNA where it's stored to the ribosome where the information is going to be read to actually make a protein. And that leads us to the second stage, which is called translation. You can think of it, the term translation means to convert from one language to another language. And that's in fact exactly what happens in this step. So the cell is going to read the information that's coded in the mRNA. This is written in the language of nucleotides, and it's going to use that information to create a polypeptide. Polypeptides are written in a completely different language, right? They use amino acids, not nucleotides, as their building blocks. So this is a process of translation, reading the information in the mRNA and using it to build a new polypeptide out of amino acids. And the translator in this process really is the ribosome. It helps to facilitate sort of this interaction between the mRNA and helps to build that polypeptide. Okay, now there are differences in gene expression between prokaryotes and eukaryotes, as you probably could have guessed. First of all, let's just generically go through this process of gene expression in prokaryotes. Remember that prokaryotes, they don't have any organelles, right? There are no subcompartments on the inside of the cell, and so transcription and translation really occur in the same space. The DNA is hanging out there in the nucleoid region. Molecules come in and help to transcribe that DNA into an mRNA. As soon as the mRNA is made, it's ready for translation. a ribosome can hop onto it and start reading the mRNA and build that new polypeptide. In eukaryotes, it's a little bit more complicated, and it's complicated because the process of transcription and translation are spatially separated. In our cells, we have organelles, and so that means we have this compartmentalization on the inside of our cells. The DNA is located inside of the nucleus, and it cannot leave the nucleus, whereas the ribosomes, they're outside in the cytoplasm, and they can't enter the nucleus. And so how do you get the information that's stored in the nucleus out to the ribosomes that are hanging out there in the cytoplasm? This is exactly where that mRNA is so important in eukaryotes, because it is that messenger that carries the information from the DNA out to the cytoplasm where the ribosome is. And so if we take a look at, again, sort of very holistically, this process of gene expression in a eukaryotic cell. So in purple here, we've got the nucleus. This is where the DNA is hanging out. This is where transcription is going to take place. So the cell is going to read a segment of this DNA for that particular gene, and it will create a new mRNA. Now, this is not a fully baked RNA. This is what we call a pre-mRNA, or on the next slide, we describe it also as the primary transcript, the initial RNA transcript that's created in eukaryotic transcription. Going back a slide again. This pre-mRNA is not ready for translation. We'll talk a little bit later about why that is, but it has to go through a couple steps of RNA processing. So once it's processed, now becomes the fully baked mRNA, and it's ready to go into translation. Only that completed mRNA has the ability to pass through the nuclear pores in the envelope and get out here into the cytoplasm, where the ribosome can then attach to it and translation can occur outside of the nucleus. All right, and so probably at some point, you've heard about the central dogma of biology, and this is the concept that cells are governed by a certain cellular chain of command. DNA stores the genetic information. That's then passed on to RNA. RNA carries that genetic information to the ribosome, where it can be made into a protein, the eventual product of the cell, right? So the central dogma, DNA to RNA to protein.